Good morning. I am Dr. Alexander Kummer from the Veterinary channel. Today, I'm going to present you with 5 questions in an effort to help you with your biochemistry studies. These questions also serve as a review for your NAVLE exam. The first question is, all of the following subcellular organelles can be isolated essentially intact except A. Mitochondria, B. Nuclei, C. Peroxisomes, D. Lysosomes, E. Endoplasmic reticulum. The answer to this question is E. Gentle disruption of cells does not destroy mitochondria, nuclei, peroxisomes, or lysosomes. However, when the endoplasmic reticulum is disrupted, it forms small vesicles called microsomes. Second question. Lysosomes may A. Secrete their enzymes from the cell for digestion of complex carbohydrates. B. Combine with mitochondria to provide additional oxidative phosphorylation potential for the synthesis of ATP. C. Combine with phagocytic vacuoles to become digestive vacuoles. D. Combine with peroxisomes to become digestive vacuoles. E. Secrete digestive enzymes to degrade microtubules for exocytosis of enzymes and hormones. The answer is C. The lysosomes combine with phagocytic vacuoles to become digestive vacuoles. Third question. Which reagent is used for the quantitative determination of amino acids in general? A. Pauli's reagent. B. Sakaguchi reagent. C. Ninhydrin. D. Periodic acid. E. Methylene blue. The answer to this question is C. Ninhydrin is the reagent used for the quantitative determination of amino acids. Fourth question. When protein subunits combine to form a quaternary structure, all of the following interactions may occur except A. Hydrogen bonding. B. A hydrophobic interaction. C. Van der Waals force. D. Disulfide bond formation. E. Electrostatic interaction. The answer is D. The quaternary structure is stabilized exclusively by non-covalent interactions. Disulfide bonds are covalent. Now to our fifth question. Proteins may be separated according to size by A. Ion exchange chromatography. B. Isoelectric focusing. C. Polyacrylamide gel density gradient. D. Molecular exclusion chromatography. E. Ammonium sulfate precipitation. The answer to this question is D. Ion exchange chromatography and isoelectric focusing separate proteins on the basis of charge. Polyacrylamide gel is used for electrophoresis to separate proteins according to size in the presence of sodium dodecyl sulfate, but not for density gradient. Molecular exclusion, also called or named as size exclusion chromatography, separates molecules depending on their molecular size by filtration through a gel. Question number 6. Black tongue in dogs is caused by a deficiency of A. Thiamine. B. Riboflavin. C. Pantothenic acid. D. Niacin. E. Alpha-tocopherol. The answer is D. Black tongue is related to niacin deficiency in dogs. Now to our next question. Trypsin digest. A. Sucrose. B. Unsaturated fatty acids. C. Proteins. D. Complex carbohydrates. E. Lipopolysaccharides. The answer is D. Trypsin digests complex carbohydrates. Question number 8. Vitamin K is necessary for A. Platelet aggregation. B. Activation of thromboplastin. C. Modification of prothrombin. D. Synthesis of hemoglobin. E. Activation of fibrinogen. The answer is C. The modification of prothrombin and factors 8, 9, and 10 to form the glutamic acid residues occurs during synthesis by a carboxylase enzyme located on the luminal side of the rough endoplasmic reticulum. Vitamin K is an essential cofactor for this carboxylase enzyme. Question number 9. As compared with non-ruminants, ruminants derive more volatile fatty acids from A. Vegetable proteins, B. Carbohydrates, C. Vitamins, D. Lipoproteins, E. Nucleic acids. The answer is B. Ruminants derive more volatile acids from carbohydrates. Question number 10. When it binds to oxygen, hemoglobin exhibits A. Negative cooperativity, B. A sigmoid saturation curve, C. A beta barrel structure, D. An alpha helix structure, E. An allosteric conformational state. The answer to the question is B. Hemoglobin exhibits a sigmoid saturation curve when it binds to oxygen. The next question is the following. When the plasma activity of an intracellular enzyme is abnormally high, all of the following are possible, with the exception of A. Tissue damage may have occurred, B. The enzyme may have been activated, C. The enzyme's rate of removal from the plasma may be depressed, D. Quantification may shield valuable diagnostic information, E. A competitive inhibitor is usually given to reduce it to normal levels. The answer is E. Competitive inhibitors are often toxic to animals. Question 13. Types of physiologic regulation of enzyme activity include all of the following except A. Covalent modification, B. Competitive inhibition, C. Change in the rate of synthesis of the enzyme, D. Allosteric activation, E. Covalent catalysis. The answer is E. Covalent catalysis involves a covalent bond between an enzyme and a portion of the substrate. This is question number 12 of our biochemistry review for veterinary students, section 1. Metal ions may A. Participate in oxidation reduction processes, B. Compete with the substrate for the active sites of enzymes, C. Serves as a rate-limiting step in enzyme activity, D. Participate in the committed step in enzyme synthesis, E. Activate enzyme degradation. The answer is A. Metal ions may participate in oxidation reduction processes. Our 14th question is, cell membranes typically contain all of the following except for A. Cholesterol, B. Phospholipids, C. Transmembrane and surface proteins, D. Free glucose or glucose attached to gangliosides, E. Carbohydrates in the form of glycoproteins and glycolipids. The answer is D. Cell membranes do not contain free glucose or glucose attached to gangliosides. This is question 15. Membrane channels. A. May form clusters that create gap junctions between two cells. B. Are closed when the transmembrane potential is changed. C. Commonly contains an amphipathic beta barrel structure. D. Has a non-selective permeable gate for cations. E. Has protein structures largely impermeable to cations. The correct answer to this question is A. Membrane channels are quite specific. Voltage-gated channels are controlled by a change in the transmembrane potential, but nicotinic acetylcholine channels are chemically regulated. The transport system that maintains the sodium and potassium gradients across the cell membrane. A. Is a symport system. B. Involves ATTPase. C. Is an electrically neutral system. D. Allows sodium into the cell. E. Requires CAMP for activation. The answer is B. Sodium-potassium ATTPase regulates sodium-potassium transport. It is an antiport, vectorial electrogenic system, and does not require CAMP. Question 17. If mitochondria are exposed to rotenone, A. 6-8 oxidation remains normal. B. The rate of NADH oxidation is diminished to 2/3 of the normal value. C. Electron flow is inhibited at the coenzyme Q level. D. Oxidative phosphorylation is uncoupled. E. Electron flow ceases, but ATP synthesis from NADH continues at 2/3 of its normal value. The answer is A. Rotenone inhibits at site 1, preventing all electron flow and all ATP synthesis from NADH. Cyanide feeds electrons at site 2 and thus is not affected. If tightly coupled mitochondria that actively oxidize cyanide are exposed to cyanide. A. Electron flows cease, but ATP synthesis continues. B. Electron flows cease, but ATP synthesis can be restored by subsequent addition of rotenone. C. Subsequent addition of oligomycin causes greater oxygen uptake. D. Subsequent addition of 2,4-dinitrophenol causes ATP hydrolysis. E. Subsequent addition of azide restores ATP synthesis. The answer to this question is alternative D. Cyanide inhibits electron transport at site 3, blocking electron flow and ATP synthesis. Subsequent addition of an uncoupled activates ATPase to hydrolyze ATP unless it is inhibited by a phosphorylase inhibitor, such as oligomycin. This is question 19 of our biochemistry review questions for veterinary students. Which tricarboxylic acid cycle intermediate may be added or removed by another metabolic pathway? A. Isocitrate. B. Oxalosuccinate. C. Alpha-ketoglutarate. D. Cis-aconitate. E. Fumarate. The answer to question 19 is C. Alpha-ketoglutarate can be formed from glutamate.